Hello everyone, welcome to the channel Microbial World. In this video, we will be learning about adaptive immunity. We will be focusing on the important characteristics, components and types of adaptive immunity. So, adaptive immunity is also called as acquired immunity. It is a specific defense system of the body. It is a type of immunity that develops after exposure to a pathogen or foreign substance. It is slower to respond initially, but it offers long-term protection and immunological memory. Let us see what are the main characteristics of adaptive immunity. Specificity. Adaptive immunity targets specific antigens. Now, how do they do this? They target specific antigens using unique receptors on B and T lymphocyte. So each lymphocyte recognizes only one specific antigen. As we already said, adaptive immunity has immunological memory. So after first exposure to an antigen, the immune system creates memory cells. So upon second exposure, the immune response will be faster, stronger and longer lasting. Diversity. This means the body can recognize and respond to millions of different antigens using vast variety of lymphocyte receptors. Self versus non-self recognition. Adaptive immunity can distinguish between our body's own cells and foreign invaders. So normally they do not attack self tissues because adaptive immunity has self-tolerance. Clonal expansion. When a lymphocyte recognizes its specific antigen, it undergoes proliferation or cloning. This produces many identical cells to fight the infection effectively. Delayed response. Now, unlike innate immunity, adaptive responses takes days to develop upon first exposure, but it is more precise. Regulation. Adaptive immune responses are highly regulated to prevent overactivation and damage to normal tissues. Now let us look into the main components of adaptive immunity. B lymphocytes or B cells. B cells are produced and mature in bone marrow. They are responsible for humoral immunity. When B cells are activated, they differentiate into plasma cells and memory B cells. Plasma cells produce and release antibodies, whereas memory B cells provide long-term immunity. As you can see in the figure, B cells have specific receptors called B cell receptors on their surface. These receptors bind to the antigen directly, like parts of bacteria or viruses. When the antigen binds to the B cell receptor, B cell becomes activated. Sometimes it needs help from helper T cell to become fully activated. Once the B cells are activated, they multiply to form many identical B cells or clones. Some of these B cells become plasma cells. Plasma cells secrete large amount of antibodies into the blood. These antibodies bind to antigen and help to destroy it. T lymphocytes or T cells. They are produced in the bone marrow and mature in thymus. They are responsible for cell-mediated immunity. There are three main types of T cells: helper T cells or CD4 positive T cells, cytotoxic T cells or CD8 positive T cells, and regulatory T cells. Helper T cells activate B cells, cytotoxic T cells, and macrophages. Cytotoxic T cells kills virus-infected and abnormal cells. Regulatory T cells suppress and regulate immune responses. Antigen-presenting cell. Antigen-presenting cells capture, process and present antigen to T cells via MHC molecules. Examples of antigen-presenting cells include dendritic cells, macrophages and B cells. Now, what are MHC molecules or major histocompatibility complex molecules? As we already said, MHC molecules present the antigen to T cells. MHC molecules are of two types, MHC class 1 and MHC class 2. MHC class 2 molecule is present only on antigen presenting cell and they interact with CD4 positive T cell, that is helper T cell. Now how do they do this? Antigen presenting cells such as a dendritic cell engulfs the microbe through the process of phagocytosis. The microbe is then broken down into smaller peptide fragments. These antigen fragments are loaded onto MHC class 2 molecule inside the antigen presenting cell. The antigen MHC2 complex is then displayed on the surface of antigen presenting cell. Helper T cells recognize this complex and bind to the antigen MHC2 complex using T cell receptor. The binding activates T helper cells. Activated T helper cells release cytokines and help other immune cells. MHC class 1 molecule is present on all nucleate cells. It interacts with CD8 positive T cells. MHC1 molecule presents intracellular antigens, for example, viral proteins or tumor proteins. The proteins inside the cell, such as viral proteins, is degraded by proteasome, and the resulting peptides are transported into the endoplasmic reticulum. MHC class 1 molecule in the endoplasmic reticulum binds to the peptides, and the MHC1 peptide complex is then transported to the cell surface. It is recognized by CD8 positive cytotoxic T cell, which then kills the infected or abnormal cell. Cytokines. Cytokines are small proteins released by immune cells to communicate and coordinate immune responses. Examples of cytokines include interleukins, interferons, and tumor necrosis factors. Types of immunity. Based on the response type, there are two types of immunity: humoral immunity and cell-mediated immunity. Humoral immunity is mediated by B lymphocytes and antibodies. It defends against extracellular pathogens such as bacteria and viruses in body fluids and their toxins. Now let us look into the steps involved in humoral immune response. Antigen-presenting cells such as macrophages recognize and phagocytose the pathogen. The antigen is then processed and presented on MHC class 2 molecule. Helper T cells recognize the antigen and release cytokines. This enhances B cell activation, proliferation, and differentiation. Activated B cells multiply and form clones of plasma cells and memory B cells. Plasma cells secrete antibodies specific to the antigen. These antibodies circulate in the blood and lymph to neutralize or eliminate the pathogen. Cell-mediated immunity. It does not involve antibodies. It is mediated by T lymphocytes. Cell-mediated immunity targets infected cells, cancer cells, and foreign tissues. As we have already said, an antigen-presenting cell engulfs a pathogen and presents antigen via MHC molecules. Helper T cells gets activated when antigen is presented via MHC2 molecule. Activated helper T cells release cytokines, and the cytokines in turn activates B cells and cytotoxic CD8 positive T cells. So cytotoxic CD8 positive T cells becomes activated when an antigen is presented via MHC1 molecule. As we said, full activation requires cytokines released by helper T cells. Activated cytotoxic T cells release perforin and granzymes to kill the infected cells. Perforin creates pores in the targeted cell membrane, and granzymes enter through the pores and trigger apoptosis, that is programmed cell death. Some T cells becomes memory cells for faster response if the pathogen returns. Based on how immunity is acquired, there are two types of immunity: active immunity and passive immunity. Active immunity develops when a person's own immune system produces antibodies or memory cells against the antigen. Active immunity takes time to develop, like days to weeks, but it provides long lasting or lifelong protection. There is natural active immunity and artificial active immunity. Natural active immunity is developed naturally after being exposed to a pathogen. For example, you get infected with measles and you're recovering. Your body now has memory cells against measles. Artificial active immunity is developed after deliberate exposure to a weakened or dead pathogen through vaccination. For example, MMR vaccine for measles, mumps and rubella, polio vaccine or COVID-19 vaccine. Passive immunity. Passive immunity occurs when ready-made antibodies are transferred from one individual to another. It provides immediate but short-term protection. There is natural passive immunity and artificial passive immunity. Natural passive immunity occurs when antibodies are transferred from mother to baby via placenta or breast milk. For example, IgG antibody crossing placenta or IgA antibody in breast milk. Artificial passive immunity is given by injecting antibodies made in a lab or taken from a person or animal. This is done when quick protection is required. For example, tetanus antitoxin taken after an injury or rabies immunoglobulin after a dog bite. So this is all about adaptive immunity. I hope that the video was useful for everyone. Thank you for watching.